camera. Good afternoon. Hello, everybody. We're broadcasting here from UCL in London. Our welcome session for the newly arrived trainees of SysMIC Module 1, an introduction to system skills for bioresearchers. I start by introducing the colleagues who have joined me here in our studio. On my left side is Geraint Thomas.
Hello.
Director of the event. And on my right side is Rob Stanley.
Hi.
Who will be one of our tutors and who will be following your chat comments. Okay, thanks for being here with us today. And let me start this session by sharing my screen and giving those of you who have not done this with the GoToTraining software a little introduction on how to use the control panel. So if you have joined us, you will have the control panel open, and there's a few things that I would like to mention. On the upper left or upper right side, there's a small orange button which you can use to hide the control panel feature, which is very useful if you need to have as much screen as possible to view the materials that we'll be showing. A bit further down is the audio section. Make sure that you have chosen mic and speakers, then you can hear everything properly. Or, for that case, if you're joining us by telephone, please check the telephone box instead. A little bit further down are materials, which are PDF files of anything that is related to the session that we do today, which by clicking on it, you can just download onto your computer and view them and use them later on if you like. At the top, no, not at the top, at the bottom, you can find a chat box. So because we have many people simultaneously watching us today, we don't allow you to show your screen and to talk with us directly, but we'll have the possibility to pick up your chat comments, and Rob will kindly be attending to those and either answer them individually or bring it to our awareness, and we will try to address it during the session if possible. If there's anything more complicated, we might refer it to later on and answer it after the session. Okay, so have a look at those materials. While we're getting ready, you can download them, and that should be all for this part. Our session will be structured as follows. After this brief overview, we'll have a poll where we'll try to find out whether everybody is set up to actually get working on the module. Geraint will then be giving an introduction to the SysMIC Enterprise, after which we have a quick test to test your knowledge. And finally, I'll give an introduction into how the training goes when you use SysMIC materials, just to give you a bit of a flavor of the whole story. If we have a couple of minutes left, we can try to pick up some chat comments, and we'll be then closing the session and hoping to see you again in the next session, which is then going to deal with the first materials, namely the introduction to the MATLAB, and that's a week from now. Okay, so I'm going to take this away, and while Geraint gets his slide ready, I take my web camera away to save a little bit of streaming power, and I'll open up for you the poll, which is an important thing. Namely, the question to you out there is, do you have MATLAB running on your computer or laptop? That means actually running, and can you actually click on the icon and open a MATLAB command window and type in commands? If yes, please click yes. If no, please click no. Because you know you won't be able to do any of the work unless you have MATLAB running. So we just want to check that we are 1 week into the course, whether everybody's really set up for this work to do. We'll wait a couple of more seconds while messages are coming in. And it looks pretty good so far. So very few who answer no. If you have not answered yet, please take the time to click now. Otherwise, I will close the poll. And I'll do this right now. And if we take a look at the results, I think this is fairly positive that most of you have been able to access the web page, to download things, and to get your MATLAB up and running. Which is good news, because it means you can start with session number 1 shortly. Right, let me close this down now. And after this short introduction, we are now going to have a quick overview. And Geraint Thomas will be guiding you through the course as it stands and as it has come about. Geraint.
Okay, thank you very much, Gerold. So this is an introduction that's very similar to one that I've been running around the country with to various BBSRC doctoral training partnerships and institutes now for the last month or 2 months or at other points in the year. And the idea is to really give you a sensation of where this course has come from, the motivations for its creation and delivery, and the sort of things that are going to feature and why they're important. So to start with, then, let's go right back to the sort of diagrams that appear in the BBSRC's strategy and policy documentation that describes exactly the sort of things that they wanted to achieve from 2010 through 2015 and then into the future. So the BBSRC has a remit to contribute to some very large activities, focusing on doing world-class bioscience, especially to be interested in bioenergy and industrial biotech, in food security, and to basic biosciences underpinning health, in order to drive us towards a good economic future with plenty of activity in the new and emerging knowledge environment, and to have real impact on the quality of life here and abroad. But all of this activity is required to take place in an environment in which there's a considerable focus on exploiting new ways of working. And that is not in trying to do science as it was done in the 20th century, but as it's being done now in the 21st century and as we expect it to be done in the future. And clearly there's going to be a greater emphasis on looking at living systems as precisely that, as systems, and to concentrate on or explore features that emerge from the combination of parts, and not just on studying the parts in isolation. The other feature that's novel in the 21st century compared to the 20th century is the fact that data is getting cheap to collect. Data can be collected and archived in vast amounts, but what we're lacking often is the talented people to turn all of that information into knowledge, and that knowledge into understanding. And in order to handle all of that data on these complicated systems, you're going to need to apply some pretty complicated tools, and many of these will be computational or mathematical, or you will need to interact with people who are deep experts in these in mathematics and computing, but be able to bring your biological knowledge to bear on the project and to help to direct it and to pay off. Now, to make this happen, there's going to need to be a very vigorous exchange of knowledge, and this is going to require you to have skills on the boundaries between disciplines. If you need to communicate with engineers, physicists, life scientists, mathematicians, computer scientists, statisticians, just about the only languages that are sufficiently compact and unambiguous to allow this to happen efficiently are the languages of mathematics. So this is why there's a considerable emphasis on this lingua franca between all of these different disciplines. And the hidden agenda behind a lot of this is that we want to reach a stage of biology that is predictive, in the same way, in many ways, that the disciplines of physics are, for example. We want to be able to have a biology that extends beyond the intuition of the individual investigator, and where there can be a little bit more certainty about our expectations of how things will behave, both from the molecular scale up to ecosystem scale. So the course was created after winning a competition by the consortium that consists of UCL, Birkbeck College in London, University of Edinburgh, and the Open University. There's very little overlap and redundancy between the competencies of these four contributors, each bringing expertise in life sciences, biosciences, or in their research and their teaching. And also because of the contributions of the Open University, especially, and also from Birkbeck, a great deal of unrivaled expertise in distance learning at the postgraduate level. To say a little bit more about this, UCL has a long history of conducting research on the life and clinical sciences interface between computer sciences, mathematics, engineering. It's been well funded here for a long time, and very large doctoral programs have been running for quite a while. In fact, Rob Stanley, who's here as a tutor today, is a member of the Complex Centre at UCL, and comes to you as a mathematician who's become deeply interested in bioscience problems. The Open University, as I mentioned, is a colossus of distance and open e-learning. The Open University have been working at distance with advanced materials and learners for over 40 years. They are, as I say, their expertise is unrivaled, even in the modern era of MOOCs. They have been working for a long time with particularly difficult materials, with students who can focus only part-time, but still work effectively. At any one time, they have many thousands of postgraduates and undergraduate students working at the postgraduate levels and undergraduate levels in mathematics, statistics, and computing. And thus all of the materials that they contributed to this course are known to work and have been refined through real life experience. Birkbeck College London have been running advanced postgraduate courses in difficult aspects of modern molecular and biological sciences, particularly crystallography and protein structure. They've been running these through the web for almost as long as there's been a web to deliver things through, and they have considerable expertise in this area. Our partners in Edinburgh are attached to Synthesis, but also the School of Informatics there have contributed significant amounts of material to the course. Synthesis is, of course, a centre that focuses on systems and synthetic biology. We want you to take on aspects of mathematics and mathematical biology and computational biology in the way that mathematicians take them on these days, not with chalk, or not with pen and paper and chalkboards necessarily, but to turn immediately to computational methods to do much of the heavy lifting for you. And the two program environments and software you'll see in this course will be MATLAB and also R for statistical work. These are very popular tools for work in this field. A particular interest to life scientists working through SysMIC, I hope, will be something irrelevant to most biologists, which will be the analysis and understanding of networks. And by that I mean the interactions of parts. A fundamental principle of life sciences is that it's not about the scale of things. Most living systems are all on similar scales, unlike things in physics that are either at the quantum level or the cosmological level. So the scale is always about the same for biology, but it's different from physics, for example, in that the complexity is much, much greater. The interaction of parts is pretty much the fundamental unifying feature of biological problems. And we want to introduce you to the fact that mathematicians have been very active in this sort of area for a long time, and there is a vocabulary and suite of ideas well developed already to allow us to understand and to analyze complicated systems. We need to introduce you, though, to some mathematics that you may have been missing. It's become apparent that in the A-level syllabus in the UK and before that the use of matrices has virtually disappeared from school-level teaching. Some of it may appear in undergraduate courses, but we definitely need to bring this dimension of mathematics into play for the work we want to do. Once you become familiar with just how powerful these approaches are, you'll wonder why they were missing from your education beforehand, and presumably be very happy to have them back as part of your portfolio. One of the things we want to do, the places we want to get you to is the idea of reaching for simulations early on when trying to understand any sort of problem. So, for example, in this slide, we've got a little genetic circuit here in which we have inducers and promoters and repressors. And the challenge is to try to understand from some sort of given starting point how the system will evolve over time and how the levels of different proteins will rise or fall. Now, these, depending on the initial conditions, the system can go to any number of different endpoints. The question is, which one will it go to? And through introducing you to simulation, we want to get you into the habit of not just guessing what the outcome of particular systems or the behaviors of systems will be, but being able to work from from a limited initial knowledge of what the system is and a little bit of knowledge about how it's starting and to predict how it's going to behave. And for that, we need to get you used to simulations. As you go through the course, you'll realize something that if you have colleagues who are engineers have known for a very long time indeed, and that is that there is a formalism for building models of mathematical models of systems. And in this example, it's shown as a cycle and going around clockwise here. But the important point here is that because it is a cycle, you can pretty much break in anywhere. So if you already have data, but you don't really know precisely what it means, then by building a model, you can help to understand what that data might be trying to tell you. You might not have any data at all. You might only have a bit of information about how you think the biological system is set up. You can create a model, a first approach model to help to refine your thinking such that you can design experiments to optimally feed into the model and go round this cycle several times until you arrive at something that better approximates or represents reality. And we want to introduce you to something that your, as I said, engineering colleagues have known for years, and which it's now important for biologists to pick up. We'll be very interested in getting your skills up for analyzing data, because at the end of this whole process, we'd like you to build mathematical models, for example, of biological systems that really can interact with data and represent the real world. We're going to give you various e-learning tools to make this happen. Here is a screenshot from a simple one in which we have a 3-gene repressolator network in which the levels of various components rise and fall in an oscillating fashion according to various parameters like initial protein concentrations that feature in the model. And we would like you to, first of all, play with representations like this to understand the power of what simple systems of equations can deliver, so that you realize that they are going to be powerful things for you, so that you'll feel highly motivated to go on and create your own and use them with enthusiasm. So to begin to start wrapping up, there's an important point here that describes the environment of the course. And that is, why should you bother to continue to improve your skills? Well, it is a fact that you need to keep developing in order to remain current. And right now, one of the most exciting interfaces that we have in modern biomedical sciences is this interface with mathematics and computing and large sets of data. So this is an ideal area in which to be adding new skills, or in which to be looking to add new dimensions to your work. So here in this little cartoon, we have a sort of common conundrum that will be facing your existing or future employers in the research sector and in other sectors. So at this water cooler conversation here, we have two executives from a company, and the first one, the chief financial officer, for example, is saying, what happens if we invest time and money in developing people and then they leave? So this is the risk for the research enterprise, both in academia and in industry. Why should you as individuals or universities or research institutes invest time, effort, and money, which are essentially all the same thing, into training when you might leave and go on and do something else? And then the chief executive says, well, what happens if we don't have highly trained people and they stay? So we can't afford to have, in our most ambitious projects and in our most exciting research areas, people who aren't prepared to really work in as a productive and effective way as possible. And therefore, everybody needs to have the best skills they possibly can to take on both the problems that are in front of them now and the problems that may emerge unexpectedly tomorrow, the week after, in a month's time, or a year, 5 or 10 years down the line. And this brings us then immediately to the real world of jobs and job applications and remaining competitive. There is every good reason to undertake training and to continually improve your skills to make sure that you are maximally competitive in the job market. And the research job market is a particularly tough one, and it's an easy one in which to get left behind unless you can add extra value to everybody's research projects. So I'll begin to summarize, and this slide is maybe a little bit condensed. This idea of marching towards predictive biology and making biology in the 21st century a predictive science is a huge undertaking. And it's not going to be left just to people who identify as biologists any longer. It's going to be fertile ground for people from mathematics, computing, statistics, engineering, all playing together to take on the enormously important problems which need solving now. And the future, as I said, relies on collaboration across these disciplines. And to be an effective worker in this knowledge industry, you need to be able to communicate what you've found to other people in languages that they can understand. And you need to be able to absorb their outputs. You need to be able to take in information in mathematical form, for example, and deploy it in your investigations of biology. And it's this exchange that will be critical. So this course, SysMIC, is a resource for you. It's for you to pick up and use as you want. The idea is that all of these materials remain accessible to you after the 6 months of this delivery has closed. And if things go to plan, it will be continually updated into the future. So these materials will not stagnate and will remain current 1, 3, 5, 10 years down the line, all being well. In addition, we hope to supplement much of the material here with new features. So bespoke courses for people working in industry, bespoke courses on good statistical practices in various research fields, for any number of different things. We intend to add new programming languages. For example, we're developing materials in Python. That once you've studied in MATLAB, it should be easy to move into this different language. But we hope, though, that the core will develop practical mathematical modeling skills, and that this will help to fundamentally change your contribution to interdisciplinary research and make you much more effective. And that ultimately it will end up, your new skills will provide crucial support to creativity and innovation in biosciences research. And I'll finish there. Thank you very much.
Okay, thank you, Geraint. So with this overview of the SysMIC course, let me take off the slides for a moment and maybe quickly I'll bring up something that was just being questions on our chat box. So there was a question on the materials. For those who cannot access the materials, here's a list given, and the materials will be available on the Moodle side of the course. We'll put them there after the session. Right, so I just tell you what is there. Geraint mentioned that we're trying to persuade all bioscientists to use a new language, and a very readable and entertaining account of the same argument is given by Jure Lazebnik from the Cold Spring Harbor Lab in this nice paper that's very readable paper, Can a Biologist Fix a Radio? And I'm afraid the answer in 2004 was no, but maybe we are much better in 2014. But I think it's now known throughout the bioresearch world that this is a true argument and that it must be taken seriously. Second, you have the information about the course, which I will go through shortly there. So for to download, to look at links and to see the dates of upcoming sessions. And then we have the slides here. You can download the slides of today's course. And finally, at the bottom, you see a slide that's called Web Session Preparation. Is a PDF file that gives you a few tips how to best prepare for a session that's going to deal with any of the assignments in the course. So if you feel you're left behind, or if you feel you're not really up to date, or if you feel questions about the assignments, the web sessions will be a way to address these questions, but the best way to be prepared is to go through these notes and make a few preparatory steps and to get the best results from joining such a session. Okay, I have also prepared for you. There's also, among the notes, there's also a calendar of the release times for this module of the individual sessions. And in agreement with this calendar, we'll have regular support seminars like this one here, just on specific assignments. And you can see the calendar of the next 3 sessions that Rob and I will be doing for you on 3rd of this. No, that's not true. It's not 3rd of December. Sorry, there's a typo here. The first one will be next week. So that's 20 or 19. I think it's 19 of November. I'll correct that shortly. And the second one will be on 3rd December, yes. And finally will be on 17th of December. And it goes in pace with the release of the materials as we go along. Similar seminars will continue this series in the new year, and you will be made aware of it via emails from our administration. And then you can have all the links, and you will be told what to prepare for this session, what to expect, and how it's going to be running. Right, so then we have covered this. Now let us go to the next point and let's do a test of your current knowledge. So in order to do a test of the current knowledge, I would like to launch the test here, and the test goes like this. Most of you will be familiar with the London Underground network, and you might have seen the map of the London Underground. It's a schematic that uses lines and colors and rectangles and squares as symbols to depict what stations are there, which connections there are, and where and from when trains go. And because we are using the language of networks in biology, we're now asking, are you aware of the individual features of a network? So, can you take a look at the question here and try to answer the question by clicking? The question is, what on the London Underground network are the edges? So typical networks have nodes and edges, but what on the London Underground are the edges? Take your pick, click on, and I can see first people are starting to click. Think a little bit about it. We'll leave it open for a few more seconds. What on the London Underground network are the edges of the network? Probably easy to find out what the nodes are, but still it's a novel concept and you might have to rethink. And I'll give 5 more seconds before we close the poll. So those who haven't clicked yet should click now. You can see we have more than 70% coming in. And while I wait a little bit longer, because there's still a lot of clicking activity going on, I might tell you that you are part of a cohort that has 318 people on this module number 1. So don't think these are small numbers of people interested in this. We have a very large cohort again. About 200 are from BBSRC-sponsored doctoral training partnerships, and the rest are postdocs and PIs and professional individuals from pharmaceutical and biotechnological industry. So a large range of people interested in doing this. And with this information, I close the poll now. Most of you seem to have submitted.
Can I just, while the numbers are coming up, can I just add something there? Is that so far over 1,000 people have engaged with SysMIC. This is really a big and very exciting enterprise that seems to have picked up real momentum. Thanks.
Okay. So let's see the score. So 72% answered correctly that the tracks are the edges of the network, and I think it's a good result. So we can see that 3/4 are up to date, but it also shows that 1/4 is not yet really into this network concept, and I think it's appropriate that the first sessions of the course will go into network, the underlying graph theory, and the way how to use software to structure and display and extract information from biological network data. Okay, let me close the poll. And having a view of us, we'll bring up the video. We are now going to go to the second presentation, where I will try to show you, to give you a flavor of how working with SysMIC is going to be for you, what experience it is, and how we think that we have found a way of more directly bringing difficult parts from mathematics, computer science, and engineering such that they are being useful for your application as soon as possible, without much going around. So I'll try to get up my presentation now. It's going to start with this one, and again during the presentation I switch off the video streaming, then we have more band power. And we start with a quote from an eminent scientist who is Leroy Hood from the Institute for Systems Biology in Seattle in the United States.
So what systems biology is about is understanding the dynamics and the nature of networks and how they create the disease.
Phenotype. Sorry to cut you short, Leroy. The last word was phenotype. So, and if this is what a systems approach is, then what SysMIC is trying to offer for you is to train you in skills and tools to understand the dynamics and the nature of networks, biological networks, and how they create the biological phenotype. So we want to help you to understand and to use appropriate methodologies to get to this point where not only you have information about the individual components of a living systems, but about the biological function. And as bioresearchers, I think function is what we are after all. So our idea of setting up the SysMIC course was that we're not starting from the mathematics. We're not starting from algebra, from graph theory or other difficult things, but we start by way of example, and that means starting from a biological example. Start with something that we hope everybody is familiar with. For example, I give here an overview using this from the second session of the course, so it's in one early in the course, that deals with how to appropriately look at large-scale data that have to do with, for example, signaling networks. So it's just one example picked out, and you know that signaling networks play an important role in many of the biological contexts. And so we pick this one example, and our way to introduce this in the course is we refer you to a current paper, something that's not been published so long time ago. In this case, the paper is from 2011. And the reason we refer to recent references is that from such papers you can download data in a digital form. So you download them, bring them on the computer, and in this case, this is a study of directed protein-protein interaction. And it's a large-scale study, and it summarizes the data in one big long table that has on the left side, as you can see here, abbreviations for the proteins or the genes, the corresponding genes in place, and a pair of numbers which tells you where the interaction starts, the number on the left side, and on the right side where the interaction ends. And so you have, this is a way of summarizing the information, and it's a very long file, much longer than the short example that you can see here. So the question is then, what do you do with this data? And we'll show you why it's very useful to have a MATLAB license. Because if you have it, you can then access the bioinformatics toolbox. This toolbox has a function called biograph, and we'll show you that with a few lines of typing. So this is your code here displayed in the center of the graph that you're going to use. And typing a few lines of code, you'll be able to ask MATLAB to display this network from the Vinayagam paper from Science Signaling 2011 for you on the screen just in front of you directly. Well, sometimes not so directly. The sad news is that maybe more than half of the time you'll probably be spending in debugging. So one of the major training will be in trying to get you over the frustration that almost nothing in programming works on the first go, but that you will have to learn to look carefully at the pieces of code. And in this example here, it's just one from a practical error that happened. It turns out that the S in layout scales needs to go, and that this is what causes the error message, which is written in red here, and which probably doesn't say a lot of, doesn't give a lot of information to those who don't know MATLAB already. So we need to do troubleshooting and find out what's going on, and then we can correct this. And what you will get is, if you execute this code, is have all the data displayed in front of you on your screen. Well, it can't possibly be the end of the work, because the fact that this is a complicated thing is something that you might have guessed before. Now we need to work with these networks, and we learn simple commands in MATLAB how to then, for example, use color to code proteins that serve certain functionalities. The example here is in yellow are from those proteins studied are those that are known to be cellular receptors. So proteins that pick up information from the environment and start a signaling cascade. And similarly, in red, we have colored here the transcription regulatory proteins, so those that are involved in gene expression regulation. And if you have this knowledge to get to this point, you're now able to ask biological questions. Means you can now use MATLAB to turn it into ways to get information that's related to your work. We don't know your specific work question, but we use an example here. So if we want to know what types of receptors target a specific gene expression regulation protein, and we have chosen the example of SMART1, then we can see there's easy filtering opportunities in MATLAB to get out all of those membrane receptors that target this specific regulatory protein. And we can pick them now nicely in this circular format. You have a quick overview of who is there. And we have done even more. If you look at the thickness of the arrow, we have introduced a measure of directness of interaction. And the thickest arrow shows the one membrane protein that is most directly connected to this SMAD1 receptor, SMAD1 regulatory protein. So not only do you get where the interaction goes by means of the arrow and what type of interaction are found in this specific data set, but you also have some candidates which you might pick out for first experimental trying. If you wanted to have some impact on this regulatory protein, this is the receptor that you might pick as a candidate. And so you have used your network biology and your MATLAB experience to get this information displayed nicely in front of your screen. And of course, you can also put this into your paper. Okay, that's a lot of things that we cannot deal with right now, that you're going to go through in the next 6 months. We hope you're going to be very successful with it. We're going to give you a lot of support in different forms. I mentioned 3 ways. First of all is you will have online forums where you can post your questions, and please use them extensively. If you have doubts or you have error messages or you don't know what to do or anything that seems strange to you, please refer to the online forums. For each session there's a forum, and our team of online tutors and the SysMIC staff will try to address this as soon as possible. And we also try to pick up questions that come in during the night or during the weekend, depending on what our schedule is. We'll try to do it. And in any case, it's a way of getting out a problem, but receiving some feedback from somebody who is knowledgeable in that area. And you can also be redirected to other forum posts. And by looking at what other people have had as a reply, you might very quickly pick up an answer that will solve your problem as well. Secondly, as we mentioned in the beginning, we are going to give online support sessions. They go with each of the assignments that you're supposed to upload. So we're going to give you exercises, examples, tricks, and hints on how to do these assignments, and we'll be able to address also individual questions during those sessions that are more direct than the chat forums, because we'll be able to interact via the web directly with you. And finally, here at UCL, we're going to organize a couple of face-to-face workshops, and the administration will send you dates around. So there will be events in various locations specifically dedicated to people struggling or working with Module 1. So these are probably colleagues of you. And we hope to see you then in either in London or in another location. If you feel free, please consider coming. Sometimes it's nice to actually sit together with the people who struggle through the same material as you do. Okay, and as a final example, because Leroy Hood in the beginning mentioned, of course, to understand biological function or the biological phenotype. And many of the examples that we deal with during the SysMIC sessions, during the online sessions, because you are now at the basic level and it's all introductory, will not really go all the way down to the full phenotype or pathophenotype if you deal with diseases. But yes, it's possible. Here's an example displayed. Here's a MATLAB code that is put on some brain graph, where an advanced form of the type of modeling that you will learn is actually then extended to not only cover a few variables, but to actually describe what's going to happen on the human neocortical surface, so all over the gray matter in the human brain. And this is sort of the ultimate goal that we want to reach for many, many research questions, namely, as you can see in the figure here, that you have a display of the anatomy, sort of the anatomical substrate of where your problem is based on. And in this case, it deals with a neurological problem. It's clinical data from an epileptic seizure that we have modeled. And you have on the left side of the screen a simulation running of this model of the human neocortical dynamics. On the right side is the original recorded clinical data, and we can compare one-to-one the anatomy and the physiology, in this case pathophysiology, because there's something wrong in this brain, of the activity during an epileptic seizure, left side in the model and on the right side in the clinical data in parallel. So the ultimate goal, and I let the video run, not only to show you the video, but also to have internal testing of the bandwidth and whether the video is transmitted in good quality, where the model on the left side and the clinical data now can be compared to. And as Geraint said, in a circular fashion, a model would have to be updated in order to get a proper representation. And it's a way of describing and of researching in biological and medical problems that we think has the future. And I definitely can say that we are not the only one, that we are joined in by the Medical Research Council and by the Engineering Research Council and by the Wellcome Trust in thinking that this is the way to go forward. And so we all encourage you to never mind a few frustrations now and then, but keep going and try to get the best out of it. As we said, it's going to be applicable to your research within a very short time. And we hope you're very successful in doing these things. That concludes my part of the presentation. SysMIC e-learning course to give you these skills and tools, and we'll have a couple of minutes for discussion. So I refer to our tutor, Rob. Has any problem have shown up during the chat that you have looked at? Anything that you might pass on to us now?
Not particularly.
Not particularly? Okay, so I give you some more information. We are recording this session here, and similarly we'll be recording all the following sessions. So if there is one of the training sessions, web sessions that you can't attend because it's in a time slot where you need to do experiments or you need to harvest crop or whatever you might want to do, you'll be able to see the video later on, or short segments of videos on YouTube, and the link will be available on the corresponding Moodle site, such that you can see the video and download the materials that go with each session as well from the Moodle page.
I did get one other question about why the help sessions after submission deadlines in some cases.
Can you say it again?
About why these help sessions are after the submission deadline in some cases.
Because I know that some of the submissions are after deadlines, but from the experience, we can see that the people who need the support most are those who cannot actually finish within the deadline. And those who have professional duties or are on traveling or somehow related otherwise. So it's for those that we most target those online help sessions. We hope that those who want to submit within the deadline and have a problem will be clever enough to use the online forums, because we'll give them the feedback there. but we think that the people who really do not get it within the deadline should not be left behind because they have passed the deadline. So we want to encourage them to say, even though the deadline is gone, there's still ways to do it, and we'll still accept submissions, and it will still be possible to get a way of guiding you through it. And that's why we're doing not all just like on the last day of the submission or so. Also, we sometimes actually give half of the solution away, so we better do it afterwards. So yes, but if there's a high demand, you might also email us and ask for more. If we don't have the time to adjust it, we might not be able to do it. But if we feel there's a certain need for further session, for additional sessions, we might poll the course. Our administrator might poll the course participants, and we might be able to add in an extra session if we feel that this should be done. So you can see technological advance helps us a lot in getting forward. And I think we are ready then for today. We have seen that all of you have MATLAB running. Most of you know what the edges of a network are, so you're ready to go into it. The first session will be about using MATLAB. The session details, I think I will publish them again because it was wrong in the previous slide, is next Wednesday, 19th of November. We'll be having a support session for the MATLAB. And I can see from the registration now that in this moment we already have 86 people have registered for this site. So there is a big demand, and I encourage everybody, please consider going to these sessions or watch the video afterwards. There's a lot of helpful tips there which are also accompanied by examples which are not in the materials that you download from the website or from the PDF. So there will be additional materials, there will be additional tricks, and there will be answers to some of the questions that we find on the forum posts of the previous courses. So hoping to cover a wide range of topics. Okay, then let me get us back. I'll stop sharing my screen, and that brings us back to our UCL studio. Geraint, thank you very much for joining. Rob, thank you very much. Also everybody else, there's other people who also helped. They're all acknowledged in the notes that you can download. I hope you're going to have a good time, and maybe see you again next week. And goodbye here from London and from UCL.
Thank you very much. Bye-bye.